Hello, everyone. Thank you for joining, and thank you, Chloe, for the lovely introduction. I will be talking to you guys a little bit about human whole genome sequencing. Um, this presentation will be divided in three parts. First, I will go over a quick introduction, followed by our project workflow, then the possible content in our bioinformatic analysis, and my assistant will be Dr. Novo, so look for him throughout the presentation. So to kick this off, what is the main function of a human whole genome sequencing? Well, the main goal is to deliver a high resolution and comprehensive view of the entire human genome. And with the assistance of bioinformatic tools, one can decipher the possible variations in the genome of a subset of samples or even a population. So with that in mind, one can answer a variety of scientific questions. And in this next slide, you have a few examples of papers generated using this amazing tool. Keep in mind that for all these three papers, the scientists use our services. And if you want demonstrations on how this tool can be applied, these are really good examples. So, well, we have already understood why human whole genome sequencing is such a powerful tool. So now let's focus on how the experiment actually works in our facility. So in this slide, we'll be able to see an overview of our workflow. So it starts with sample preparation, sample quality control, library prep, library quality control, sequencing, data quality control, and lastly, the bioinformatic analysis. So let's take a deeper look into each one of these subsets, starting by the sample preparation. So we have different requirements depending on the pipeline your sample will go through. And it ranges from 1 microgram of fresh tissue to 200 nanograms of extracted genomic DNA. Make sure you have a good idea of the sample requirements before you ship them to us. And remember that we are here to answer any questions that you might have at any time. Here, I'm also showing you a few examples of the most commonly sent tissue samples, blood, oral swabs, or even cell pellets. Okay, so next in line, we have the sample quality control. In this part, we measure three parameters of your samples. We measure the concentration, the integrity, and purity. Based on the results of the qubit and the agarose gel electrophoresis, we'll issue you a sample QC report. A quality control report, which it's just going to tell you if the testing samples meet the requirements for the library construction or not. And based on that, you will be able to choose to move forward with the samples we received or ship new ones. On the picture, you have an example of three different samples QC and also the gel electrophoresis results. So once we receive the confirmation from you that we can move forward, your samples will go through library preparation. In this step, the genomic DNA is randomly fragmented by sonication to the size of around 350 base pairs. Then DNA fragments are polished, A-tailed, and ligated with the full-length adapters of Illumina sequencing, followed by further PCR amplification. The PCR products, as the final construction of the libraries, are purified with the NPure XP system. These libraries are checked for size distribution by the Agilent 2100 bioanalyzer and quantified by real-time qPCR to meet the criteria of 3 nanomolar. So to summarize, we will use qubit to measure the concentration of the library. Once this is done, we'll issue a library QC report. And we will wait for your confirmation to move on with the samples or not. Once you do so, once you confirm that you want us to move on, we will sequence your libraries using NovaSeq 6000, and the sequencing strategy is going to be PE150. If your samples passed our quality control, we also guarantee a Q30 of above 85%, and we will follow the sequencing depth established in the initial quote. When the sequencing is done, we will also perform what we call the data quality control. In this step, we will discard any contamination, low quality, and uncertain reads in order to make sure that all the downstream analysis are done using only high quality data. You can see in the pie chart that the great majority of reads in this example are clean. Also, it is pretty common that the data provided by us actually exceeds Illumina's official guarantee of Q30 above 85%. In this example, it was around 92%. So to summarize, we have really good quality data. So what are the options from there? To answer the question, we will need to go through a little bit into the analysis content of our most common bioinformatic pipeline. Well, if you remember, I said that all the downstream processes done in Novogene is performed with clean reads. And the first thing that we do is align those reads to the reference genome, in this case, the human genome. After that, we'll analyze any possible variation found in our data, and if available, we can even compare data between tumorigenic samples versus normal samples, which culminates in our final analysis, which is the annotation. Now, let's take a quick deeper look into the first part of the analysis. The first part of the aligner is utilized to map the and clean reads to the human reference genome, HG38, and the results can be obtained in the band format. As an example for this case, you can see in the graph on the left, where you have the average sequencing depth per chromosome represented by the bar plot, at the same time that you can see the proportion of covered bases by chromosomes by the dot line plot. To summarize all the data acquired, we can issue you a table with all the information obtained in the mapping, coverage, and depth measurements. So further into our analysis are the detection of single nucleotide polymorphisms, insertions, and deletions. For that, the haplotype caller is used to perform variant calling to identify any of the mentioned variations. And we also provide you with a list of what was found, as well as graphs with the number of variations found in the genomic regions, as well as the different types of mutations found in the coding regions, since those are the main target of studies. As an example, on the picture, the pie chart on the left represents the number of SNPs in various genomic regions, whereas the pie chart on the right represents the number of different types of SNPs, specifically in coding regions. We follow the same exact trend for insertions and deletions. So on the left, you have the data for SNPs, and on the right, you have the data for insertions and deletions. Lastly, in our standard analysis, we have the detection of copy number variants, structural variants, and annotation. Copy number variants are genetic variants that lead to variation in copy number of relatively large fragments, usually longer than 50 BP between individuals. There are two types of copy number variants: gains and losses of copies, obviously. CNVs may be and may form the underlying genetic basis of individual differences and cancer. You can see the table provided by us with the data of our experiment on the top left. Underneath that, we have the structural variants part, in which we follow the exact same trend as CNVs. These are genetic variants with relatively large size, including deletions, duplications, insertions, inversions, and translocations. SVs are the underlying genetic basis of individual differences and have the potential to affect even tumorigenesis. On the bottom of the left side, we have an example of annotation results. Following genetic variation detection, annotation of variants will be done by the tool called NOVAR. Immutable aspects are performed, so including but not limited to protein coding changes, genomic regions affected by the variants, allele frequency, deleteriousness prediction, and even more. So the list of databases that we use is long, so don't hesitate to contact us for more information or questions. As you can see in the example, the annotation information includes 5 parts: chromosomal regions and genic structures related to the variation, database annotation, functional prediction, and basic information on the variation and gene function and pathway annotation. Lastly, on the right, you have circus, which is a nice tool to visualize the genetic variations and the variants on the genome scale found in your experiment. So, after all of this information, is there anything else you should expect from our company? And the answer is obviously yes. We offer a variety of customized bioinformatic options, so please contact us if you have interest or have any questions. So This picture is just an example of the different types of advanced analysis for human whole genome sequencing. Our cancer advanced analysis is always very sought after by our clients, and we have had a lot of CRISPR-Cas9 on-target and off-target detection projects lately since it has become more and more common, the technique. So this is going to be the last picture, and I'm just going to leave it here to demonstrate what is done in our advanced analysis for cancer. I'll leave it right here, and I'm going to say goodbye for now. Thank you for the participants and for listening, and Somi will take it from here. Thank you.
Thank you, Mario. Hello, everyone. Thank you for joining us today. My name is Soumi Joseph, a technical support scientist at Novogene. So for this session, I will be talking to you about eukaryotic mRNA-seq sample preparation, how to get a qualified total RNA sample. Before going to the talk, I would like to give you a brief outline what I will be talking. So first, give you a short introduction on eukaryotic mRNA-seq. And second, we will look into the methods, how to check the quality of the sample, what are the sample requirements, and we'll give you short guidelines how to prepare a good quality RNA for eukaryotic mRNA-seq. And finally, look into some of the questions that we usually get it from the clients during sample submission. So, as we know, there are three major molecules in the cell, that is DNA, RNA, and protein. These are the molecules which determine the whole integrity of cell. The process that converts the DNA to messenger RNA to protein is a foundation for how cell function. Here, the intermediate messenger is the messenger RNA. So when we have the information about messenger RNA, you will get to know all the genes functionally present in the cells, the proteins expressed in the cell. Thus, if you do eukaryotic mRNA sequencing, what you are getting is the complete gene expression pattern, and also you will have information on gene function and gene structure. To give you more efficient and more standardized, a high quality service, we have come up with a smart solution that is our Falcon NGS platform. It is a fast, flexible, and intelligent multi-product, fully automated delivery platform for high throughput NGS services. It can avoid manual error during the NGS operation, starting from sample preparation all the way to BA analysis. It can process 3,000 samples per day, which can reduce significantly production cycle by 60%, and thus you will get reduced turnaround time for your service. So why do we do eukaryotic mRNA-seq? mRNA-seq has a wide range of application across different research fields, including medical research and agriculture research. In cancer research, many oncogene properties are related with the gene fusion. To understand the prevalence and the structure of the gene fusion associated with the different cancer studies, cancer diseases, the mRNA-seq is one of the best tools. And also it can discover single nucleotide polymorphism, that is SNP, which can be used as a strong molecular marker in genetic disease and breeding studies. And also in drug target studies, mRNA-seq can differentiate the gene expression pattern in a drug-treated sample versus untreated sample. And evolutionary study, clinical diagnostics, and disease-related gene analysis, in all these cases, mRNA-seq is a very valuable study tool. In agriculture research, mRNA-seq has been used to study evolution and domestication of different crops. It can identify many candidate genes responsible for important crop traits like grain size, drought tolerance, and kernel oil. So it has wide, strong application across study designs. We get a lot of projects on eukaryotic mRNA-seq. So when we get total RNA from you, the first step is we look at the quality of the sample. If it is everything is good, we will go for the next step, the library construction. But if it fails, we will ask you to resend the sample to ensure the good quality output. So here you can see typical library structure. So where RNA will be converted into cDNA, the cDNA fragments will be flagged by read 1, read 2 sequencing primer, which will use to read your left and right fragment reads. It will be flagged by the indices I7 or I5, which can differentiate different library. And finally, it will be flagged by P5 and P7 at the end, which will be bound to the flow cell of the Illumina platform. So once the library is made, we will look into the library quality. If the quality is good, next step is going for sequencing. For mRNA sequencing, we use NovaSeq 6000 sequencing platform using paired end 150 sequencing strategy. When sequencing is done, the raw data will go thorough data quality control. So where you will get a clean reads filtered from all the low quality reads and adapter sequences. So this will be ready to go for bioinformatic analysis. And if you want us to do bioinformatic analysis, we have a very good package, quantitative and standard package based on your research needs. You can, if you want gene expression analysis or structural analysis, you can choose different packages, and we will be happy to help you to choose the right one for according to your research need. So let's look into sample quality. So how do we check the quality? We have two labs at Davis and our global sequencing center at China. So in Davis lab, we use qubit to test the RNA quantity, and Agilent 2100 to check the integrity and purity, whether the RNA is degraded or not. So it's very important to check the integrity. We used Agilent 2100 Davis lab. In China lab, we use nanodrop to test the RNA quantity, and always Agilent 2100. And also we have Agilent 5400. So it's the fragment analysis for high throughput quality check. So if you want us to extract the RNA instead of sending the total RNA, we are ready to help here. So you can send cells or fresh plant tissue or animal tissue depending on your research need, but we require some standards to make sure the samples are, the RNA sample that we extract will be good enough. So if we are sending cells, so we require at least 5 million cells. If it's a whole blood, at least 5 ml for any library, if it's a non-directional or directional library. So if it's a fresh or plant or animal tissue, we need 500 milligram and 300 milligram respectively. So based on the sample source, our requirement varies. So you can reach to us and make sure all the sample requirements are right before you send the sample. So if we are planning to send the total RNA for the mRNA sequencing service, based on the sample type, we require a different amount. So if it is for non-directional mRNA library, we ask for at least 400 nanogram amount of RNA with the volume 20 microliter, with the 20 nanogram per microliter concentration. So if it's a directional mRNA library, we need a little more higher amount, around 800 nanogram, because of the processing. During the library preparation, we may lose more RNA, so we make sure we have enough for starting. So if it's the total RNA from blood type, you always have 800 microgram at least too. So in case you don't have enough RNA, we understand that always not easy to extract the RNA as you want from different tissues, but we have for different pipeline for low input and ultra low input mRNA library preparation. So for low input, you can go up to 100 nanogram RNA. For ultra low input mRNA library, you can go for 10 picogram. Always you have to make sure the ring value is greater than 6.8 for animal and 6.3 for plant or fungi organisms. So you have to make sure no degradation or contamination in the RNA to make sure we will be able to get a good quality output data. So now I would like to give you some few guidelines how to prepare a good mRNA-seq. So we have been looking into different projects that failed and successful, and we did some research to understand what makes the low quality samples. So we found that Novogene's low quality RNA is mainly due to insufficient amount of RNA or degrading RNA. So 60% of the cases was insufficient amount of RNA, 30% was due to degradation, and other 10% was due to protein contamination or DNA contaminant with other organic solvent. So now, how we can keep a good quality RNA? So thumb rule 1 is prevent degradation caused by exogenous RNase contamination. To isolate intact high-quality RNA, it is very essential that RNases are not introduced into RNA preparation. RNases are everywhere, so all surfaces, including pipettes, bench tops, glassware, and gel equipment should be decontaminated with RNase decontamination solution. For RNase-free tips, tubes, and solutions should always be used, and gloves should be changed frequently if you think it is contaminated. And always make sure you should use short operating time, so that way you can avoid all the chances of contamination and also degradation. So secondly, degradation can happen due to endogenous RNases. So while preparing your RNA samples, so you have to make sure you should use the right method. So for example, if you are crushing the tissue, so you should use on dry ice before crushing, and you under liquid nitrogen, you should always crush the tissue. And also use more lysate for during the homogenization. That way you can release more RNA and extract high output. So once you extracted the RNA, so before you have to make sure that the RNA is stored well. So before sampling, you should always confirm that amount of tissue you are using for extraction is right enough. If you start with the low amount, your output will be low, but if you overload your RNA column, then the quality will be low. So always make sure the correct amount of tissue is used. You can refer to our sample requirement table to understand, to have an idea of how much amount you should be used. So after extraction, you should use up RNA in a right temperature, especially if you're long storage, you should use -80. You can use small aliquots to store for storage to avoid frequent freeze and thaw cycle to avoid degradation. So now every cells or every tissues have different RNA abundance. So this gives you an idea how much you should use if the tissue to start with. So if you have a high abundant RNA, you can start with the low amount, but if it's a low abundant, you have to use high in material to start with. So for fresh animal tissue, the liver, spleen, or pancreas will be high abundant in RNA, whereas bone or fat tissues are very low in RNA. So in the case of plant tissue, if you want to get easy way to get the RNA, you go for blade to get good amount of RNA. So seed and flower will be very low in RNA. So choosing the right tissue organ or tissue will be a thumb rule to increase the total amount of RNA. So if you want to send the tissues to us, you should make sure that the tissues are preserved in the right method. So if you can send animal, plant, or blood or cells. So we prefer for animal and plant tissue or cells always snap frozen method. And if it's blood, you can use vaccine tubes or snap frozen, but we do not recommend use use RNA later to dissolve the tissue, because we find that most of the time it leads to RNA degradation. So finally, once everything is ready, your precious samples are ready. So the last step, but most important, is shipping, to make sure your samples arrive in our facility safely. So you have to well arrange your sample shipment. You have to always use appropriate packaging, use the parafilm to seal each tube, and place the tube in a container. And also you can use organizer if you have more samples to send, and always seal the boxes in a Ziploc bags for safety. And many times the shipping can be delayed for many reasons. So always use enough dry ice. We recommend at least 10 lb for overnight shipping. We have seen that the samples arrived very late, 3 to 3 days delayed, and most of the samples were failed. So make sure your packaging is proper, making sure they arrive in our facility safely. So finally, we'll discuss some of the questions that we usually get it from the clients. Can we accept extracted RNA in RNA later? The answer is yes, we can accept it, but we do not recommend to do that way, because we need to purify the RNA from the RNA later again before going to the QC step and downstream process. In that, we add extra cost, and also you might lose your precious sample during the purification process. Can we accept RNA samples dissolved in ethanol? Yes, we can accept it. Again, we need to do purification prior to sample QC, and then it again cause extra charge for purification and also cause great loss of your sample amount. Can we accept RNA sample preserved in RNA stable or gentograph RNA? Yes, we can do that. We resuspend them in buffer or nucleus-free water, and then do the quality check before going to downstream process. So we can accept them without any extra fee. Does Novogene charge if the RNA sample concentration is too high and needs dilution? Yes, we accept high concentration. We do not charge extra if we need to dilute them, but we would like to communicate about this before sample arrival. So with this, I would like to conclude my presentation here. I'm sure you will have more questions, and we are happy to help you. And so I'm handing over this presentation to the next speaker, Uma. Uma, this is for you. Thank you.
Hi, thank you, Sami. And thank you all for joining today, this afternoon. So my presentation is going to be on the non-coding RNA sequencing. And this presentation has 4 parts. First, we're going to introduce you to the non-coding RNA sequencing, and then discuss a little bit about project workflow, and then a detailed analysis content, and a brief case study. So let's get started with the introduction. So we all are very familiar with messenger RNA, which is the RNA that codes for protein. However, a large proportion of the total RNA does not code for protein and is called as the non-coding RNA. The non-coding RNA is actually classified into 2 groups: the housekeeping, which constitutes the ribosomal RNA and the tRNA, and the gene regulation non-coding RNA or regulatory non-coding RNA, which includes the long non-coding RNA, circular RNA, and small RNA. Right here on the right side, you can see that the total RNA, about more than 85% is constituted by ribosomal RNA, and mRNA is like about less than 10%. And then of that, the long non-coding RNA and other non-coding RNA is a very, very tiny fraction. So why is this important? Why should we study long non-coding RNA? So even though the long non-coding RNA constitutes only a very small proportion of the total transcripts, they play a really important role in many biological processes by regulating gene expression, both at transcriptional level in the nucleus and at the translational level in the cytoplasm. So as a result, they have attracted tremendous research interest. So they can compete for the messenger RNA or interact with each other and get involved in regulation of gene expression, and as a result, contribute to the phenotype. Importantly, non-coding RNA are differentially expressed in a tissue-specific manner during disease condition. So therefore, they serve as excellent biomarkers for ongoing diseases, such as cancer and cardiac disorders, aging, etc. So here at Novogene, we provide 3 types of non-coding RNA sequencing services. This includes the ncRNA-seq, small RNA sequencing, and circular RNA sequencing. In addition to the sequencing, we provide an analysis package which includes quantification, function, strand orientation, differential expression, discovery of novel non-coding RNA, the biological pathways where they are playing a role, and also regulatory relationships to target messenger RNA for non-coding RNA. And these are the applications where it can be used. So here's the project workflow. So as previously discussed, our starting material is RNA sample, and then the first step is sample QC. They go on to library preparation. Then we have a library QC step. The libraries are then sequenced, and the data is then again QC'd, and only the clean data is entered into bioinformatics and analysis. So I'll get to little specifics about non-coding RNA in terms of sample requirement. They're slightly different from messenger RNA sample requirement. The requirement for long non-coding RNA sequencing is at least a minimum of 500 nanograms. We need more for circular RNA-seq and small RNA-seq, greater than 2 micrograms. Likewise, the RIN value has to be higher for the small RNA sequencing. The materials we use include blood, cells, tissue. And as always, if your RNA does not meet this requirement, do contact us for details, and we might be able to provide you some alternatives. The next step is after the RNA is the library preparation. And the library preparation for long non-coding RNA-seq is a strand-specific library preparation with ribosomal RNA depletion. So as a result, we can get data from both strands of DNA. For circular RNA-seq, the library preparation is very similar to long non-coding RNA-seq. In addition, there's a step of linear RNA digestion. For small RNA-seq, we have a step of isolating small RNA before library preparation. And the sequencing strategy is P150 Novogene for both ncRNA-seq and circular RNA-seq, and SE50 Novogene for small RNA-seq. We can also sequence small RNA in P150 and trim the reads. Recommended data output is 12 GB for long non-coding RNA-seq, about 8 GB and more for circular RNA-seq, and 10 to 20 million reads for small RNA-seq. So next, let's move on to the analysis content. So the analysis content is pretty comprehensive for long non-coding RNA-seq. So in this presentation, we will cover only the analysis pipeline for long non-coding RNA-seq. Do call us if you need more information for small RNA-seq and circular RNA-seq. So this slide shows an overview of the analysis pipeline. The first question we ask in this analysis is, what long non-coding RNA sequences are present in your sample. Next question we ask is how many are present? And then we ask what is their biological function? And then we ask what's the target messenger RNA? This also includes comparing between groups and figuring out differential expression analysis. So as we discussed earlier, raw reads are cleaned up and only clean reads enter into analysis after data QC. The first step is mapping of the sequences to a reference genome. And then there's a step of long non-coding RNA identification. And then we also identify novel genes. And then we quantify the transcripts. We can do differential expression analysis when we are comparing groups. And eventually we do functional analysis. Now let's get to the details of this analysis package. So we discussed this slide earlier, in which we show that only clean reads enter our BI analysis. And Novogene guarantee is that Q30 is greater than 85%. The next step is mapping. So mapping is essentially mapping the sequences to the chromosome of the reference genome. And since we have a standard library prep, we can map it to both the strands. And here in the green and orange is the distribution of read density to the chromosome. We also give you a pie chart, which just tells the distributions of reads in the known types of RNA, where this pink reads are mostly messenger RNA, and the long non-coding RNA as the orange, and the different RNA, all the RNAs that are sequenced are, the distribution is shown in this pie chart. So next, after mapping, our next step is to identify the long non-coding RNA in your samples. So for this, since our information we have contains both messenger RNA and long non-coding RNA, the map reads, which is the BAM files for each sample, will be assembled into transcripts based on known gene annotations. Then the assembled transcripts from all samples are merged, and then we use additional filtration step to provide you the long non-coding RNA classification. And here is the pie chart telling where these long non-coding RNA sequences, the distribution, how they're distributed and where they are present. So as far as long non-coding RNA sequencing is concerned, a number of them are still not annotated. So we can provide you, we can make novel gene predictions from the data. So most RNA-seq studies will reveal novel genes and transcripts. And once those novel genes are predicted, we'll add those to the gene list and provide you with a table which contains the information about the novel genes and the annotated ones that are provided here. This will help you to annotate the reference genome better for your future studies. The next step is quantification of the long non-coding RNAs present. For this, we determine the read counts. The read counts can be affected by many reasons, such as how many reads are sequenced for each sample, and other factors such as the gene length. As a longer gene will lead to more reads. So we compare the data between genes in each sample across the sample. We normalize the read count for each gene by what is called as FPKM, or fragments per kilobase of transcripts per million mapped reads. So what it takes into account is that it takes into account of the gene length as well as total reads that we have. So it's the most common method for estimating gene expression level, which takes into account the effect of both sequencing depth and gene length. And then we provide you tables of FPKMs of long non-coding RNA as well as mRNA in each sample. So now we have quantified the long non-coding RNA. The next step for us is to predict the target. What are these long non-coding RNA potentially binding and affecting gene expression? So to do this, we evaluate potential co-expression between long non-coding RNA and the messenger RNA using Pearson correlation. A positive correlation between a long non-coding RNA and an mRNA is defined as a Pearson correlation greater than 0.7 and a p-value of less than 0.05. Now, this analysis can be classified into two groups as LNC RNA that cis-regulate messenger RNA or trans-regulate messenger RNA. The cis-regulated ones are the ones that are present in the same chromosome in less than 30 kilobases away from the target gene. The trans ones are those that are present in a different chromosome or farther away from 30 kb of the same target gene. So we'll provide you these results. Now we might have, in your experiment, you might have more than one group. So we can also do differential expression analysis for one or two or more groups and provide the data as volcano plots and cluster heat maps. The next step is an enrichment analysis of the differentially expressed genes. The genes that have changed, either upregulated or downregulated, we determine using gene ontology or the KEGG pathway. We determine what the function of those genes could be and what biological pathways they might be falling under. And we can also provide this as a scatter plot of the top 20 significantly enriched genes and the KEGG pathway. The last part of the analysis is a protein-protein interaction analysis. So in this, we extract the protein-protein interaction information of targeted gene list from string database and then the source database and import into software called Cytoscape and provide you data which looks like this. So here in this, each of the bubble represents a transcript. And if there's any connection between the two bubbles, it indicates potential interactions between those two transcripts. So with this, I've described the analysis content of our ncRNA-seq analysis. And if you need more information for small RNA and circular RNA, please contact us. The last part of my talk is going to just show you a case study where researchers have used long non-coding RNA-seq to look at the effect on aging. So in this study, researchers have looked at total RNA sequencing, which includes long non-coding RNA-seq and mRNA, at different stages of the age groups of mice and looked at different tissue. And this is the study design, and you can always look at the paper in more detail. And the bioinformatics analysis included quantification, differential expression, functional enrichment, and co-expression. So the last slide here shows that co-expression data, where we're looking at the interaction between long non-coding RNA and mRNA sequences. And as you can see, this is the heat map, which you don't have, which is pretty dense. But if you look at this, the figure E, you can see that the connections between the long non-coding RNA and mRNA increased as at a later time point in the age. And this interaction was highly increased in the adipose tissues. Also, there was a strong correlation to inflammatory response gene pathway, suggesting that the interactions drive inflammatory responses in the adipose tissue are associated with aging. So in this case, it can also serve as a biomarker for aging. So with this, I conclude my presentation, and please post your questions on the chat. And if you have any further questions, do not hesitate to contact us at this information provided. Thank you. And from here on, Yumi will take over. And please go ahead, Yumi. Thank you very much.
Thank you, Uma. Hi, everyone. Thank you for the informative presentations, first of all, Mario, Sumi, and Uma. Hi, everyone. Welcome to this webinar section, 10x single-cell RNA sequencing from sample to data analysis. My name is Yun Mei, and I work in the tech support team at Novogene. This presentation contains 4 parts: introduction, project workflow, data analysis overview, and a brief case study. First, introduction to 10x single-cell RNA sequencing. I wanted to start off by talking about the differences between traditional bulk RNA and single-cell RNA sequencing. I like this analogy from 10x genomics and will borrow it today. Bulk RNA sequencing measures the average gene expression levels in a group of cells, much like comparing different batches of fruit smoothies that were blended from many fruits. Single-cell RNA-seq, on the other hand, measures gene expression levels in each cell, similar to characterizing the flavors of each fruit without blending them. Therefore, bulk RNA-seq masks cellular heterogeneity, while single-cell RNA-seq is ideal for revealing cell-to-cell variation on a transcriptome level. Our 10x single-cell RNA-seq offers 2 solutions: whole transcriptome profiling and antigen receptor sequencing. In whole transcriptome profiling, either the 3' or the 5' of each transcript is captured and used toward counting gene expression levels. It's worth pointing out that 10x single-cell RNA sequencing only sequences a portion of each transcript, in contrast to traditional RNA sequencing that captures the full length of the transcripts. This pipeline is useful to identify rare cell types, to understand cellular heterogeneity, to atlas and characterize complex cell populations, and to discover new biomarkers. The second solution is antigen receptor sequencing in B or T cells. This product is ideal for profiling immune cell repertoires, determining antigen specificity of B and T cells, and characterizing tumor microenvironments. To carry out single-cell RNA-seq, we follow the standard protocol during the entire workflow. The top portion of the slide shows the main steps. Briefly, dissociated cells are loaded onto a chromium controller for single-cell partitioning. Then the samples go through library construction, sequencing, data processing, and analysis. The bottom half of the slide shows the main function of the controller. Within the controller, barcoded gel beads are mixed with cells, enzymes, and partition oil to form gel beads in motion, or GEM. Each GEM functions as an independent reaction compartment where mRNA transcripts within a single cell are converted into cDNA and are uniquely barcoded. Central to the 10x single-cell technique is the gel bead. Shown on this slide is a gel bead for 3' single-cell assay. Each gel bead is coated with primers that contains a common 10x barcode, and this barcode varies from one gel bead to another. Immediately downstream of the barcode is a unique molecular identifier used to mark reads from PCR duplicates. At the 3' end of the primers are sequences that will anneal to transcripts to initiate cDNA synthesis. Now I would like to move on to part 2, our project workflow. This slide recaps the main steps in our workflow. From our clients, we would need cell or tissue samples properly prepared. Once the cell or tissue samples arrive, we will take care of the rest of the steps. Here, I would like to point out that this workflow does not apply to nuclei samples. Please let us know if you need to submit nuclei samples. For single-cell RNA sequencing pipelines, we can process single-cell suspension, including cryopreserved cells, fresh tissue, and frozen tissues. And I'd like to also add that we can offer dead cell removal if that's needed for your samples. Next, I would like to briefly talk about the library structures for single-cell RNA-seq. Shown on this slide is a single-cell 3' gene expression library structure. On this library, the 16 base pair 10x barcode and 12 base pair UMI will be sequenced in read-1 cycles, and the transcript will be sequenced in read-2 cycles. We use single indexing strategy. Therefore, each library will have 4 8 base pair I7 indices. For downstream analysis, at least 28 base pairs of read-1 and 91 base pairs of read-2 are required. The single-cell 5' gene expression and VDJ enriched libraries are constructed from the same kit. This kit takes advantage of the fact that the V, D, and J segments of the B or T cell receptors are located near the 5' end, and VDJ sequences are enriched using sequence-specific primers. So what's the difference between single-cell 3' and 5' gene expression libraries? As their names reveal, they capture different parts of the transcripts. Within the VDJ pipeline, we offer a few library options. You can request TCR or BCR libraries separately, together, or with 5' gene expression. Regarding sequencing, we use our NovoSeq 6000 paired end 150 platform, and the recommended depth is 100,000 read pairs per cell. You will notice that these read lengths are longer than the library requirements, and we can trim the reads bioinformatically for free. In part 3, I would like to show you our data analysis pipeline for single-cell gene expression. For data analysis, we use Cell Ranger from 10x Genomics. Shown on the left of the slide is our gene expression analysis pipeline. After raw data generation, low-quality reads are removed during QC, such that only clean reads are used for analysis. Similar to bulk RNA-seq, these clean reads are mapped to a reference genome, and gene expression levels are quantified. Nextly, highly variable genes are computed. These highly variable genes are genes with very different expression levels from cell to cell within the sample, and these are used to compute principal components. One of the important goals of single-cell gene expression studies is to identify subpopulations or clusters of cells. After cell clustering, we report marker genes with variable gene expression levels among different clusters. And lastly, we provide annotation of these marker genes in a few databases. This table summarizes data QC metrics, including data output and Q30. One of the important metrics here is sequencing saturation. This value gives you an idea of roughly what percentage of the transcriptome was sampled. The mapping table provides a summary of the reads alignment outcome, namely the fraction of reads mapped to the exonic, intronic, or intergenic regions. In this table, we list various of quantification results. Some of these values are included in the web summary files from Cell Ranger. I would like to add that we also link the web summary files for each sample in our report. This slide shows the cell subpopulations or clusters using both the t-SNE and UMAP techniques. To report marker genes differentially expressed among cell clusters, we summarize them in a table, as well as showing them in a heat map, a violin plot, and in t-SNE and UMAP plots. And on the right side of the slide, a heat map showing marker gene expressions in different clusters is shown. To provide functional analysis of the marker genes, we annotate them in the Go, CAG, Reactome, and the transcription factor database. Shown on the right is an example of the top 20 significantly enriched Go terms displayed as a scatter plot. The sizes of the dots indicate the number of marker genes found in each Go term, and the color of the dots correlates with the p-value. The last part of my presentation is a case study that showcases how this technique can dissect complex biological systems. In this Cell paper published in 2019, the authors attempted to explain how stress induces anxiety on the molecular level. This study utilized a lot of different molecular biology and genetics techniques, and we will focus on the 10x single-cell RNA sequencing part. To start off, previous studies have suggested a link between persistent anxiety and the left amygdala region of the brain. The authors asked a simple question: which cells in the left amygdala are responsible for anxiety-like behavior? To address this question, they isolated single cells from the left amygdala of wild type and a genetic model of anxiety and subjected them to single-cell RNA sequencing. In the left figure, the authors show that cells from the left amygdala can be nicely grouped into 8 clusters on a t-SNE plot. Prior to this experiment, the authors have already demonstrated that a biological molecule called xanthine from T cells is linked to stress-induced anxiety. As you will notice in the right figure, one of the xanthine receptors called ADOR1 transcript is enriched in a group of cells called oligodendrocytes and OPC, or oligodendrocyte progenitor cells. And these are marked with green and blue colors in the left figure. Now looking at the left side of the picture, comparing the top 2 plots of the left figure, using a mouse model for anxiety, the omega-2 deficient mouse, the authors show that the number of oligodendrocytes, which are the green cells again, significantly increase in the omega-2 knockout mouse. And this increase is reversed by removing CD4 plus T cells as shown in the bottom plot. The right figure shows the CAC enrichment analysis of differentially expressed genes between the wild type and knockout animals. And the authors found the pathways that may be responsible for oligodendrocyte overproliferation in response to xanthine. Using other techniques, the authors already demonstrated that physical stress increases the level of xanthine in CD4 plus T cells, which enhances proliferation of oligodendrocytes. Now, using 10x single-cell RNA sequencing, they were able to establish that oligodendrocytes may be the cells responsible for anxiety-like behavior, thus completing the molecular pathway of how stress induces anxiety. In summary, with our 10x single-cell RNA sequencing service, we present 2 solutions: gene expression and immune profiling. We streamline our services to take this process off your hands. Additionally, we offer in-depth data analysis using the manufactured software packages. Lastly, I would like to thank you for joining in on today's webinar. If you have any other questions or project needs, please don't hesitate to email us or submit an inquiry on our website. With that, I would like to end my presentation and hand it back to Chloe. Thank you for your attention.